Nobody's doing anything today, it sounds like. How was the exam? Too hard? What's that? What? That's why I ask you, man. Is it too hard? What do you think? Too easy? Fair? Okay. You know, nobody ever says it's too easy, and I always find that kind of surprising. You know why? No. I mean, the best thing that you want at this point in a curved class is a hard exam, because that's what spreads everybody out. And if you have an easy exam, everybody is bunched towards the top, and your good grade may not be enough, right? So in a curved class, you always want a hard exam. But I will never, ever hear a class say it was too easy, because they're used to a 90, 80, 70, 60, right? So in a 90, 80, 70, 60, easy is good, because easy gets me up into that 90 to 100 range. But what if the class average is 90 and you made an 89? An easy exam just got you a C. Ugh. Right? That makes sense? Now, we don't need to talk about grades at this point. But tell me about the content. Somebody said it was fair. That's always good to hear. But then I never know it's because I'm standing here glaring at you or what. So is that what you expected? No? Okay, that's fine. What did you not expect? Right. Who said no? I like honest people. Okay. Yeah. You're prepared for something a lot worse. So urban legends are out there, aren't they? So I'm glad that you didn't find it as bad as you thought it was going to be. Okay. The exam is not yet graded. The TA thought she would have it graded by today, but she tells me now she thinks she'll have it tonight. If she gets it graded tonight, I will bring it to class tomorrow, and I'll pass it out in class. That's where we'll be. Yeah. I'll post a grade distribution, everything, so that you'll see exactly where you are. You'll say, oh, I made a 42, that was an A. Right? Which, if you made a 42 and you made an A, right? That's not going to happen. But I mean, my point is, a curved class is not the same as a 90, 80, 70, 60, 50, right? Everybody's kind of nervously looking at me. Average in my class is typically about 65 to 75, somewhere in that range, just like any other class. So the urban legends really aren't true. Question? I'll post the key, yes. The key will be posted outside my office. I always wait until we're giving you the exam back before I post the key. And the reason for that is once I post the key, if you don't have the exam in your hands, oh my God, I did it, right? If you have the exam in your hands, you have a known thing there, right? So the key will be posted on the bulletin board outside my office as I pass the exam out. Okay. I hope you all did well. I'd be very happy. Okay. And it's an important subject. It's a curved class. Okay. Well, let's put it this way. If everybody made over 90, I would give everybody an A. How about that? Would that be fair? For the term. Yeah. If everybody made 90, I would give everybody an A in the class. That's correct. Now everybody's going, yeah, it'd be easy, right? Don't listen to that bullshit. Right. Okay. So, well, I hope everybody did well. As always, if you have any questions, please come see me. I'd be happy to meet with you and talk with you about the class or about the material, etc. What I want to do today is spend some more time talking about control of enzymes. And so control of enzymes is important because for the example I gave the other day, we don't want to be taking a Maserati at 200 miles per hour to Fred Meyer to get our groceries because it's not a good career move for all involved, including me and my Maserati. So I want to be able to control that Maserati so that I'm going at a reasonable speed for the environment in which I'm planning to take that Maserati, right? And similarly, I need to control my enzymes. So I showed you last time how covalent, I showed you a couple things. I showed you allosteric control and how allosteric control uses molecules to bind to enzymes to either activate or inactivate those enzymes. We can turn them up, turn the volume up. We can turn the volume down. Okay. I talked about covalent modification. And covalent modification is a very important consideration. I started telling you about phosphorylation and dephosphorylation, which is a way that some enzymes are controlled. I showed you feedback inhibition. where an entire pathway is controlled by controlling one enzyme. It's a very, very common mechanism for controlling entire pathways. Control one enzyme, you control everything. Okay? Well, I spent just a minute talking, or a couple minutes talking about zymogens, and I want to say just a little bit more about zymogens. So zymogens, you will hopefully recall, are inactive forms of enzymes that are made by cells. And the example I gave to you were the proteolytic enzymes that are made by your pancreas. And I pointed out that if you had them activated in the wrong place, they could attack the pancreas and cause a very painful and in some cases life-threatening disease known as pancreatitis. So pancreatitis is a very graphic example of problems with enzymes active where they should not be. There's another place where regulation of enzymes by covalent modification, that is zymogens, is very, very important. And this one I find incredible, to be honest with you. Okay? Your bloodstream is flowing through your arteries and veins right now, and you're not even thinking about it. And it's there, it's doing its job, and if you go out and you are putting away your books and you slice your finger with the edge of a piece of paper, okay, you will notice a painful experience for a minute or two, and you won't bleed to death, which is really a remarkable thing. And you take it for granted that you don't bleed to death because, well, A, you've never bled to death, because if you did, you wouldn't be sitting here. But B, you don't even have to think about it. Your blood supply carries in it the ability, okay, to stop its own flow within seconds. And it does that because it's carrying around inside of it, all the things necessary for forming a clot. Not only that, but that stuff that's necessary for forming a clot is not routinely forming a clot. Because if it did, you would have a stroke or a heart attack. Okay? Not only that, but it's capable of forming that clot at the site of a wound. Not randomly, but only at the place where you have a wound. It's able to make a protein polymer called fibrin, F-I-B-R-I-N. Fibrin is the active form of the clotting protein. The inactive form of fibrin is known as fibrinogen, consistent with the names we've seen before, the O-G-E-N on the end of it, indicating it's a zymogen. Okay? Now, it turns out that there's a whole cascade of inactive forms of enzymes involved in the clotting scheme that get activated to form other enzymes that are activated, that form activated forms of other enzymes. So we start with a very small signal, and then at each level, the activation of one kind of enzyme amplifies the signal, and that amplified signal then activates the next level of enzymes, which increases the amount of signal. And this happens so rapidly that within seconds, we actually start forming a clot when we injure ourselves. The last thing that's amazing about this clot is it's watertight. It's watertight, folks. You never thought about it. You only notice when you have that scab on your hand after you have cut yourself. But you took for granted that you had a clotting system that did that. That clotting system is full of zymogens. As I said, the end product is something known as fibrin. And then on top of that, the body has a way of dissolving that fibrin as you heal, so that you don't have a clot sitting on your skin for the rest of your life from a place where you cut yourself. It's incredible. It's really incredible. That illustrates, I think, as clearly as I can tell you, the importance and value of zymogens. Okay? Now, the clotting system just does sometimes go awry in the clotting in our blood supply, and people do sometimes get a stroke because of a clot that forms in a place where it shouldn't. But that's the rule, not the that's the exception, not the rule. Question? So a scab is almost purely fibrin. Yes, it's a polymer of a protein. Good question. So when you form a scab, you may have noticed this if you've ever played with your scabs. When you form a scab in two levels. You form something called a soft clot and a hard clot. Okay? So the very first thing that forms is called a soft clot. And I could show you, and I'll just briefly tell you since you've asked the question. The soft clot is a polymer of fibrin, okay, that interlocks, and it is, as the name would suggest, soft. It barely holds water. You ever notice a clot as it's forming? It will be kind of fluid, and you may see stuff coming off of that while that is forming. It becomes as what you described as brittle over time. So what's the difference between a soft clot and a hard clot? In a soft clot, you have no cross-links between the individual fibers. No cross-links. So cross-links are chemical bonds, covalent bonds that form between individual fibers, not unlike we saw in collagen. Okay? As those cross-links form, okay, then the clot starts to stiffen and becomes brittle. That brittleness is important for the long-term stability of the clot and also preventing any liquid loss as a result of that. Pretty cool. Other questions? Okay, so that's what's happening in the clotting process. All right, well, I said there was a third way that enzymes are controlled, and I'm not going to talk about it here. I'll actually talk about it briefly later in the term. But the third, I will remind you, is the third mechanism by which enzymes are controlled is the way in which enzymes are either made or not made, that is, synthesized as proteins. If they're not being synthesized in a particular cell, then that, of course, is the ultimate way of controlling a particular reaction, and in many cases, a complete pathway. If one enzyme is missing, then the whole pathway may be blocked. Well, the last thing I want to talk about relative to the control of enzymes is the mechanism by which enzymes act. And I will tell you up front that I'm not a big person on mechanism. All right? I think that I will tell you a little bit about mechanism, and believe me, there's a lot of complexity. There are entire courses taught on enzymatic mechanisms. I have a colleague who spends most of his biochemistry class, in fact, talking about biochemistry, about enzymatic mechanisms. And for our purposes, I don't think that's important for us, so I don't go through that in much depth. But we should understand some basic principles by which enzymes work, and that's what I try to focus on here. Let's talk about chymotrypsin. Chymotrypsin was that protease that we talked about before that was synthesized originally as an inactive form known as chymotrypsinogen. And chymotrypsin is synthesized by your pancreas, and chymotrypsin can, in fact, be involved in pancreatitis, as I talked about, if it's activated too soon. When we study chymotrypsin, we discovered something interesting. So to explain what you see on the screen, I've got to tell you a little bit about biochemists. Okay? Biochemists are basically lazy people. Okay? Like everybody else, we don't want to work any harder than we have to. And we like to, when we collect data, have data that's coming in a relatively simple fashion and an easy-to-analyze fashion. If I want to study a protease, for example, I have to start with something that is a peptide bond that the protease can break. And determining if that breakage occurred or not can be a very complicated thing. It's not a trivial analysis. So biochemists, because they're lazy people like me, okay, have made various reagents that look like peptide bonds and that fool the enzymes enough that the enzyme will treat it as if it's a peptide bond. Well, why do I care about this? One of those reagents that we make is this mouthful of a name. I don't even have it on here. Here it is, paranitrophenolate. You don't need to know that, okay? This is a reagent that looks to chymotrypsin like it's a bond that it can cleave. Okay? And chymotrypsin will actually break it, and when it breaks it, it converts the molecule from being colorless to having a yellow color. So I can actually study the kinetics of this enzyme by following the appearance of color. Okay? And if I do this in a graph like this, what do I see? Well, this looks to you as, oh, it's a V versus S plot, except for it's not. right? This is the concentration, I'm sorry, this is the amount of product released. So that's velocity, very much like what you saw. But instead of plotting substrate concentration, we're plotting time. So if we look at the amount of color released over time, we see something interesting with chymotrypsin. We see that it has a biphasic mode to it. A rapid burst, and then a more slow phase that looks like this. And when people saw this for the first time, they realized that chymotrypsin was actually going through two steps in its catalytic process. A fast phase that corresponded with this part, and a slow phase that corresponded with this part. Now what I'm going to do is I'm going to take you through the mechanism, and I'm going to show you the part of the reaction that's fast, and then I'll show you the part of the reaction that's slow. Okay? Everybody with me? Okay. Let's take a look at this reaction. Here's our enzyme. Here's the molecule that gets fooled, that fools chymotrypsin. Here's the product. You don't need to know these structures. Don't worry about them. You don't need to know the names. Don't worry about them. But the important thing is that a molecule is made that has a color, and that color is yellow. Okay? We see this broken down into two steps. We see a first step where the enzyme becomes covalently linked to part of the molecule. This is a covalent bond. That E stands for enzyme. And this enzyme is covalently linked to part of the substrate. That's the first step of the reaction, and that's what we call the fast phase of the reaction. The second step in the reaction involves the release of the covalent substrate from the intermediate to release this guy over here. This is the slow phase. Now notice we start over here with free enzyme, and we ended up down here with free enzyme. The enzyme ends up the way that it started. Because if the enzyme didn't get freed of that bond, the enzyme would catalyze exactly one reaction. Question? So the enzyme is getting linked to part, this part right here, of the substrate. This is the substrate right here. Part of it. The other part is released. Now I'm going to show you the mechanism how that happens. Everybody understand that? Now, for the first time, we are seeing the enzyme that is making a covalent bond with a part of the substrate. Okay? This is an important aspect of this enzymatic reaction. A covalent bond that's transient between the enzyme and a part of the substrate. I'm not going to do that. Let's go here. Okay? Let's not even do... Okay, let's do that. Let's do this. When we look in the enzyme, what we're doing is we're actually looking, this is the enzyme itself. These are the bonds involved in the enzyme. And I've drawn, or somebody has drawn, some of the atoms of some of the side chains within this enzyme. You can see the side chain of a serine. You can see the side chain of a histidine. And you can see the side chain of an aspartate. Okay? What's the charge on aspartate? Physiological pH. Side chain. Negative. It's an acid, right? What's the charge on a histidine at physiological pH? It's positive. Okay? And what's the charge on a serine at physiological pH? 0. But what is notable about serine? It has an OH group. Okay? Now, all three of these things play a role in the catalytic mechanism of this reaction. These three enzymes are not close in primary structure. What does that tell you? We're in tertiary structure because when we put things together that are not close in primary structure, we had to have had folding occur, and therefore we're talking about tertiary structure. So these guys, you can see, a really good example of folding. They're not close in primary structure, but they're very close in physical proximity, meaning that they've been put close together as a result of the folding process. Okay, very good. These three amino acid side chains are what we call the catalytic triad. The catalytic triad. This catalytic triad occurs in many proteases. Chymotrypsin is one of them. Let's look at how this reaction occurs. Now I've drawn these guys where I'm focusing on the histidine and the serine. We can imagine that over here on the right, we're looking at an aspartic acid. All right? So let's go through this, and I'm going to take you through the steps, and I'm not going to expect you're going to draw this for me. Okay? You're not going to draw this for me. I want you to understand it. All right? So, to orient you, serine with its OH, histidine here, aspartic acid somewhere over here. Here's the substrate. What is the substrate? Well, the substrate is a polypeptide chain that is bound in the active site. That's true for any enzymatic reaction. The substrate will always bind in the active site. So this substrate is a polypeptide chain. It is bound in the active site. What happens when something binds in the active site? What happens to the enzyme? Changes shape. All right? There's a slight change in shape of the enzyme as a result of the binding of the proper substrate in the active site. That change of shape is critical for everything that happens here. Everything. Okay? When this enzyme changes shape very slightly, what happens? Well, that aspartic acid that's there is negatively charged. Correct? And that negatively charged aspartic acid sitting over here sees this ring of histidine over here. And the ring is something we call an electron sink. It's full of electrons. See the double bonds? Everybody see the double bonds, right? Now, I lied to you just a little bit. This guy isn't positively charged yet. It's sitting here without the extra proton on it. And meanwhile, it's full of electrons. And meanwhile, this negative thing over here has just come in close to it because a slight shape change occurred. What do you suppose the electrons in this ring are going to do? Well, if I'm an electron, I'm going to move over. I'm going to avoid the negative charge that's over here from the aspartic acid, and I'm going to move over here. So that means this side of the ring is going to be more negatively charged than it was. Okay, everybody with me? The shape change that brought the aspartic acid in also brings this guy closer to the serine. So the three have gotten more scrunched together. And look at this guy. It's very negatively charged. It sees this proton sitting over here, and what's the charge on a proton? +1. This guy actually pulls a proton off of serine. Thus, histidine becomes charged +1. And the reason it's pulling it off is because of this rearrangement, the electrons, the charge, and hey, there's a proton. I'm a bully. I'm going to grab the proton. That, okay, is what starts the entire process happening. What's left behind? Well, we have something left behind that's an oxygen with a negative charge. Because the proton left its electron behind. This thing didn't need any more electrons. It left the electrons behind. The ring is positively charged. The oxygen on the serine is negatively charged. It's something we call an alkoxide ion. A-L-K-O-X-I-D-E. An alkoxide ion is extraordinarily reactive. Extraordinarily reactive. And what does it do? Well, it's something we call a nucleophile. What did you learn about nucleophiles in organic chemistry? They what? Nucleophiles love nuclei. Okay? They have electrons because this guy is already negatively charged. So it's looking for a nucleus. And the closest nucleus to it turns out to be this carbon in the peptide bond. This carbonyl carbon is electron poor because this oxygen here is pulling electrons away from it. This guy's got plenty of electrons, just looking for something that's missing electrons. It attacks it. So the alkoxide ion attacks the carbonyl carbon, And when it does that, it breaks the bond between the carbon and the nitrogen. We're seeing that actually happening right here. This attack, you see now there's a transient bond that has occurred. And the upshot of that attack is that there's a covalent intermediate between part of the peptide chain, and the bond is broken, so the rest of the polypeptide chain is free to go. Breaking that bond splits these two guys right here. We have just completed the fast phase of the reaction. The fast phase of the reaction is done. Repeating, okay? Binding of the substrate, rearrangement of the catalytic triad, attack of the hydroxyl by the histidine ring, formation of the alkoxide ion, attack of the peptide bond, release of half of the polypeptide chain. That all happens very, very quickly. Now we see we have a covalent bond between the polypeptide chain and the enzyme. We've got to get that polypeptide chain free of that enzyme, or the enzyme is dead in the water. Okay? A couple things happen. Here's that bond. So we're basically, let's see, make sure I orient right here. We are basically right down here. Okay? We've gone from there down to here. Okay? Notice what has happened next. You see this guy right here? It's water. Water has entered the active site, and it's going to play a role in the next step of the reaction. This takes a while to happen. It's why this is so slow that there's a second phase to the enzymatic reaction. Water comes in. Exactly the same thing happens that happened before. But instead of taking the proton off of serine, it takes it off of water. Okay? That makes a reactive OH. The reactive OH attacks this carbon for the same reason that the alkoxide ion did. And now this gets released from that serine. Now the second polypeptide chain is free to go, and the reaction is done. Questions right here? Yes? So the histidine actually is not taking a second proton. It gave up a proton in the process of resolving this structure. So it's a good question. But it's actually back to having a charge of +1 when it takes that second proton. And it gives that proton up, ultimately, in making this structure right here. Yes, Mariah? So the active OH rate, this pulls a proton, which creates a reactive hydroxyl, which is what this is right here. It attacks the carbonyl carbon, just like the alkoxide ion did. And in this case, it breaks a bond between the oxygen and the carbon, so that now this guy is free to be released. Okay, so my first question to you is, could you tell me that in words? Maybe, maybe not. I see that expression on your face, right? Would you like me to review all the steps for you? Okay. Let's start at the top. Okay? I'm going to give you a couple names in the process that I didn't give you during the mechanism because I didn't want to throw too many names at you. The enzyme binds the substrate in the active site. The enzyme has a specific name for that binding site. It's called the S1 pocket. So the first step in the process is the enzyme binds the substrate in the active site S1 pocket. Okay? Second step, enzyme changes shape. Third step, the change in shape causes histidine to take a proton from serine, forming the alkoxide ion. I'll slow down. The alkoxide ion is reactive. It attacks the carbonyl carbon of the peptide bond and forms a bond with it. The other half, the polypeptide, is released. Fast phase is done. In the slow phase, water invades the active site. Proton gets removed from water by histidine. Reactive hydroxide attacks the carbonyl carbon and releases the second polypeptide chain. Pretty cool. This guy right here, okay, that intermediate is pretty unstable. The reason that the bond breaks here is because this bond is very, very unstable. It's so unstable that the enzyme could actually react with that. If it reacts with this unstable bond, the enzyme is going to be dead in the water because it won't be able to resolve that. So the enzyme has another chamber in the active site that protects the enzyme from having that reaction occur. Okay? Another chamber. That chamber has a name of its own. It's the last name I'm going to give you here. It's called the Oxyanion Hole. Oxyanion. The Oxyanion Hole is a stabilizing chamber in the enzyme. If you don't know anything else, know that the Oxyanion Hole is a stabilizing chamber of the enzyme. It's protecting the enzyme from reacting with this unstable molecule. Okay, questions? That's a complicated mechanism. That's why I don't talk too much about mechanism. I really could spend the whole term talking about mechanism if I started doing them one at a time. Now, why do I show you this mechanism? I show you this mechanism because, first of all, I told you that other proteases commonly have this same catalytic triad and exactly the same mechanism. Notice that the thing that is the most reactive here is that serine because it forms the alkoxide ion. Enzymes that have this architecture of serine, histidine, aspartic acid are called serine proteases. Chymotrypsin is a serine protease. Trypsin that I've talked about before, that's a serine protease. It uses almost exactly the same mechanism as chymotrypsin does. There are many serine proteases that are out there. You learn one mechanism, you've learned them all. You say, I'll never think about this after this class again anyway, right? It's a joke. Yes? The catalytic triad will always be serine, histidine, and aspartic acid. Yes? And a catalytic triad is always present in a serine protease. Exactly right. Okay? Everybody with me? Kevin, you lied. That's not a catalytic triad, but this is also not a serine protease. There's something called a cysteine protease, and I show it to you because now that you've hopefully begun to understand a serine protease, I'm going to show you that a cysteine protease does pretty much the same thing. It just does it with different amino acids. The serine protease had serine, histidine, aspartic acid. The cysteine protease has histidine and cysteine. Okay? Cysteine differs from serine in having an SH side chain instead of having an OH side chain. It's an enzyme called papain. It's found in papaya. Okay? I'm going to tell you, and you only have to write this down because it's going to hopefully make sense to you. All right? How does a cysteine protease work? Well, first thing that happens is the enzyme binds the substrate in the active site. That causes a slight shape change to occur in the active site of the enzyme so that the histidine is brought into close proximity with the cysteine. And the cysteine, which has relatively large numbers of electrons over here, pulls off a, I'm sorry, we're doing it up. The geometry isn't quite right, but this guy pulls off a proton from the sulfur that's on the side chain of cysteine. And that creates a reactive sulfur, just like we had an alkoxide ion. What does that reactive sulfur do? It's a nucleophile. And it attacks the carbonyl carbon, just like the alkoxide ion did. And everything else that we saw in the cysteine, in the serine protease happens in the cysteine protease. We have a fast step. We have a slow step. We have a covalent intermediate. We have water coming in the slow step to release the covalent bond. Everything is the same. The only difference is we have a sulfur instead of having an oxygen, and we don't see an aspartic acid down here because this enzyme doesn't even need it. Everything else is the same. Yes? The other enzyme used the aspartic acid because it helped with the distribution of electrons to get that up here. It turns out that oxygen actually holds on to its proton more tightly than sulfur does. So this proton is easier to remove. It doesn't take as much pull, as it were, which is why this doesn't need the aspartic acid. Question? Oh, okay, I thought it was a hand. Now you know about cysteine proteases. That's cool. You say, what did I learn today? I learned about cysteine proteases and how they're like serine proteases, right? Okay, how about we stop talking about mechanism? Spend just a minute talking about things that help mechanisms to occur. Okay? In the case of serine and cysteine proteases, we saw that the things that did the reacting were largely amino acid side chains. The side chain of an oxygen, the side chain of a sulfur, basically played those important processes. I've talked about coenzymes and cofactors before, how they help a protein to do what they do. Remember heme? Heme was a cofactor. It was a prosthetic group that helped the enzyme to do its thing. Prosthetic groups allow enzymes to expand their repertoire of reactions that they can catalyze. These guys here are all coenzymes or cofactors. You can call them either. I don't care. Okay? They help enzymes to do what they do. And if you look at these, you will notice that many of them are vitamins. Biotin is a vitamin. Okay? Flavin coenzymes, vitamins. Lipoic, that's misspelled. Lipoic acid is a vitamin. Okay? Nicotinamide, also known as niacin, is a vitamin. Okay? Vitamins are found in trace quantities because they're only needed in trace quantities to go into some enzymes to help them to catalyze reactions. Deficiencies of any of these vitamins have severe consequences. Now, I'm not giving you this table to have you memorize a table. Thiamine, there's another one. Okay, pyridoxal phosphate's another. Okay? But there are a couple on here I'm going to show you because we're going to have you know because we're going to talk about them later. One is this guy right here, biotin. Biotin's very important because it's always involved in enzymes that catalyze what we call carboxylation, putting a carboxyl group onto something. Carboxylation. Okay? Coenzyme A, we're going to see later, is involved in fatty acid oxidation. It carries fatty acids as they're being oxidized. Okay? The flavin and nicotinamide coenzymes are both involved in oxidation reduction reactions. I think I'll stop it there. Okay? Those enzymes we'll talk about later in the term because of the importance that they have for metabolism. Here's NAD, nicotinamide adenine dinucleotide is what it stands for. NAD is probably how you'll know it. NAD, okay, has a structure like you see here. It's called nicotinamide adenine dinucleotide. There's the nicotinamide part. These are both nucleotides, okay, both nucleotides. And they're joined by this phosphate bond right here. Nicotinamide adenine dinucleotide. That's what that looks like. Okay. Now, this guy is involved in oxidation reduction reactions because it can carry electrons. Carrying electrons in metabolism is absolutely critical because cells can't have electrons floating around willy-nilly. Right? If I light a match, right, and that flame starts burning, oxidation is going on. And there's a real downside to that oxidation going on there, right? It's a little higher temperature than I could have if I had the same oxidation going on in my cells. My cells would burn up. Biological oxidations are very controlled. Okay. Another problem with the willy-nilly release of electrons is that electrons as they're released cause reactions to occur that cells can't control. They cause reactions to occur that cells can't control. Cells, I've said it before and I'll say it again, cells are control freaks. The more they can control their environment, the less likely they are to have damage. And the more things are out of control, like reactions that are not under the guidance of an enzyme, the more likely the cell is going to die. By controlling electrons, cells are controlling reactions. And that's what's happening with electron carriers. This is an electron carrier. The flavin nucleotides are also electron carriers. What does an electron carrier mean? Well, we're looking at that nicotinamide part of that molecule from the last figure. Okay? We're not going to worry about the mechanism, but I want you to see that we're starting with this guy over here. Okay? This is a resonance structure, which for the moment we won't even worry about. But if we take this guy and we add a proton with 2 electrons, we create this guy. Notice the difference in structure between this and this. This guy has gained a hydrogen. It has gained 2 electrons. Notice we have 1, 2, 3 double bonds there. And notice we have 1, 2 double bonds there. Gaining of electrons, because see the electrons next to that hydrogen, gaining of electrons involves a reduction. Loss of electrons involves an oxidation. If this guy goes this direction to the left, that's an oxidation. If we go this reaction to the right, that's a reduction. Now, cellular oxidations release electrons, and these guys grab them. Cellular reductions require electrons, and these guys donate them. Now, I'm going to reiterate that when I come back and talk about some specific oxidation and reduction reactions later. We call them oxidation reduction reactions or redox reactions because every reduction is accompanied by an oxidation. If I oxidize molecule A, I'm taking electrons away from it. And those electrons don't disappear. They go to something. They reduce molecule B. Right? Every oxidation causes a reduction of something else. We can see this guy is getting reduced coming in this direction, which meant something else got oxidized moving to the right. If we take these electrons and give them away, we end up with this thing over here. Something had to accept those electrons. It's getting reduced as this is getting oxidized. Okay, vitamin B6. Memorize all those structures, right? No. All right. And I'll talk about that later. Okay. That right there is what we'll talk about with respect to control of enzymes. everybody looks tired. Is everybody tired? Do you want to stop here and then cover more tomorrow? That always is the option, right? If we stop here today, we'll cover more tomorrow. And then tomorrow's like, oh, my God, I didn't mean that yesterday. I don't want to do that, right? All right. Well, how about we sing a song? This one I want to have some help with because I don't sing it very well. But I'll help sing it. I hope you will, too. It's an old Beatles song. I know my music is old, but let's see. There we go. I answered 3B, but then I thought it might be C. Or was the false true? I can't undo. It makes me blue. It asks me to list all the enzymes that regulate fat. As I wrote them down, I discovered I didn't know jack. I ought to give thanks, scoring some points, filling in blanks. I squirmed in my seat, feeling the heat, shuffling my feet. Jump too soon. Professor then told me there wasn't a chance I would pass. So I started crying and fell through a big pane of glass. I suffered no harm, 'cause I awoke to my alarm. Oh, nothing compares to deadly scares, student nightmares. And yes, I still have those dreams just like you guys do too, so